So as you can see, the parasympathetic and parasympathetic and sympathetic nervous systems regulate similar effectors, but what is not shown here is why they would elicit their effects. There's a fine balance between the two, and by working in concert with the endocrine system, they produce homeostasis for our bodies. So we can see here that the parasympathetic nervous system controls our pupils, stimulates saliva, constricts bronchi, but then you look at the sympathetic and it does the opposite. Instead of constricting the pupils, it's dilating them. Instead of stimulating them, the saliva, it's inhibiting saliva. Instead of constricting your lungs, it's relaxing them. Slowing the heartbeat in the parasympathetic, accelerating it. Remember that sympathetic nervous system that we talked about was fight or flight? All of these make sense. When you have to get up and go because a lion's chasing you, not super realistic, but your pupils dilate so that you can see more stuff. More light can come into your eyes so that you can react quicker to things that are coming in. You don't need saliva when you're running, so you don't need that. It relaxes your bronchi so that you can breathe more efficiently. It accelerates your heartbeat so you get more blood pumping, more oxygen to your muscles. So it's all these things. Well, you don't really need that when you're in the parasympathetic nervous system. When you're not being chased by a lion, you can constrict your pupils, have saliva, constrict your bronchi, slow your heartbeat, stimulate peristalsis and secretion for your internal organs for digestion, digest and rest and digest. All right, we'll break these down a little bit further here. So parasympathetic nervous system, its effector organs are the same as the sympathetic. So either smooth muscle, cardiac muscle, or glands, and that's what we see here. The regulation, just like the sympathetic, is involuntary. These are under normal conditions, non-hyperactive conditions, non-stressful conditions. So when you're just kind of chilling. Your duration will be unconscious and continuous. So it's just happening as long as we're in a normal state of homeostasis. Relaxation, food processing, and energy absorption are all qualities associated with the parasympathetic nervous system. And the maintenance functions of it, just like I was saying before, are rest and digest. When you're relaxing, your body isn't under any stressful conditions. It can enter a different metabolic phase and allow digestion to occur, resting your body for when you actually need to be stressed, and in an emergency situation like the sympathetic nervous system. Effector organs, the exact same as the parasympathetic, smooth muscle, cardiac muscle, and glands, just the opposite effects. The regulation is involuntary, similarly, but under emergency conditions. The duration is still unconscious, yet continuous, and this is involved in tissue metabolism, alertness, and intense physical activity. And you'll see this when we're talking about fight or flight reactions. So should I peace out of here and run away, or should I fight? And both of those require similar actions for smooth muscle, cardiac muscle, and glands. So here's a nice little summary with another perspective on the peripheral nervous system. Your key point here is going to be your effector organs. For the somatic nervous system, skeletal muscle is going to be our effector organ, while with the autonomic nervous system, it'll be smooth muscle, glands, cardiac muscle. And the key difference between the parasympathetic and sympathetic nervous system are the neurotransmitters that are released that cause either an inhibitory or stimulatory effect. So they affect the same areas of the body, but they put out a different chemical which elicits a different effect on those same organs. So for instance, we can look at the stimulus of a bear, kind of scary for a human. Well, the peripheral nervous system with sensory receptors and nerves will sense that, conduct action potentials from the periphery to the central nervous system, which is the brain and spinal cord. The CNS will process and integrate this information and initiate responses to carry out mental activity as well as physical activity. So they send those on to the somatic and autonomic nervous systems. The somatic nervous system affects skeletal muscle so that you can run away. The autonomic nervous system affects your cardiac muscle, so your heart pumps faster, getting more oxygen to your body and nutrients for your muscles and your mind. Your smooth muscle to stop digesting. I don't need to worry about digesting food right now. I need to run, and also smooth muscle like lining your blood vessels to dilate so that you can have more blood flow, as well as your glands like your adrenal gland. I need immediate epinephrine so that I can get going, adrenaline, right? So it'll affect those glands. And so all of these things are working in concert. You're working with your peripheral nervous system and your central nervous system. We have different divisions, but they work in concert to maintain homeostasis so that you can stay alive when you're running from a bear. Because that happens a lot. Okay, so neurophysiology. Obviously, we've been talking about neurophysiology the whole time, but now we're going to talk about specifically neuron homeostasis. So we'll talk about the resting membrane potential, what that means, how we get an action potential. We'll touch on a graded potential, and we'll talk about synapses and how they're important. So the electrical state of the cell membrane can have several variations. These are all variations in the membrane potential. A potential is just a distribution of charge across the membrane, and it's measured in millivolts, or MV. The standard is to compare the inside of the cell, the cytosol, to the outside of the cell, the extracellular fluid, so that the membrane potential is a value representing the charge on the intracellular side of the membrane based on the outside being 0. And so we see that here in the electrode that's recording the intracellular environment to be negative 70 millivolts, and it's comparing that to the reference electrode in the extracellular space. So this is negative 70 millivolts compared to the extracellular fluid of being 0. Okay? The slight difference in charge between the two environments makes the membrane polar. It is different in charge, so there is a polarity to the membrane. The polarity creates a charge disparity, and it drives neurons and muscle cells to generate electrical signals, including action potentials. But before we start talking about those electrical signals, we have to understand what the resting state of the membrane is like. And so what we see in the resting neuron is a disparity of the ion distribution. There are more negatively charged ions or molecules inside the membrane than outside, and there are more positively charged ions or particles outside than in. Notice that there are more potassium ions on the inside of the cell and more sodium ions on the outside of the cell. This disparity is important for generating electrical signals, or action potentials. And so our resting membrane potential is negative 70 millivolts typically on the inside compared to the outside, and there is a polarity, a difference in charge between the inside and outside. And so when we're talking about an action potential, we're talking about a rapid change in the axon membrane eliciting an electrical charge or electrical signal or nerve impulse down the length of the axon. Okay, it's a wave of depolarization down the length of the axon. And what I mean by depolarization is it's becoming less negative inside the cell than outside, changing the physiology of the axon, which changes the electrical signal going down the length of the axon. And so what I'll have you do is, go into the slides and watch this video explaining an action potential, and then come back to your notes of what I'm going to talk about, breaking the action potential down further. This just gives you a different dimensional mode of learning of what an action potential is, other than me telling you what it is. This is straight out of your textbook, and I highly recommend watching it, and make sure you understand it before you start trying to understand like synapses and things outside of that. So at step 3, when we're talking about the action potential, let me rewind real quick. So this is what happens when we have an action potential. We're at negative 70 millivolts for our cell, and what's going on is the cell is in a resting membrane potential, but a stimulus makes a change in the cell membrane, the axon membrane, such that sodium channels are going to open. Sodium begins to enter through those channels into the cell. That makes the inside of the cell more positive, thus changing the membrane potential in millivolts to go up. So it's not negative 70, it's going closer to 0 or plus 40. So it's going up. So at 0, sodium channel is closed, but the stimulus opens it, sodium ions enter. So that's 1, we're here. At 2, potassium channels are opening, and potassium begins to leave the cell. So that's kind of counteracting the sodium entering the cell. Regardless, sodium channels become refractory at a point when the cell becomes so positive. Basically, voltage-gated channels that are only opening in response to the voltage being low sense that it's changed to a higher voltage, so they close. And that's what it means by refractory, means that they close, which means sodium can't enter the cell anymore, which means potassium is still continuing to leave the cell, causing the membrane potential to drop again, go back closer to -70. And that's exactly what we saw. We saw the refractory of the sodium ion channels occur, and so it stops entering. And what we see as potassium continues to leave the cell, it becomes more negative, returning the membrane potential back to resting level. Potassium channels eventually close, and so do the sodium ions reset back to closed, and extra potassium outside diffuses away, and we return back to a -70 millivolts. So this action potential gets propagated down the length of the axon. All right. So this is basically everything I just said summarized. The video did a nice job summarizing the action potential, but we can go ahead and break it down a little bit. To start, a stimulus of some sort needs to initiate the sodium channels to open, letting sodium in. The stimulus will elicit those sodium channels to open and allow the positive sodium ions to enter, which causes a depolarization, meaning that the membrane is becoming depolarized or less negative. It's closer to equal charge inside and out. So it's depolarizing. The interior of the axon becomes more positive. Potassium gates open, letting potassium out, which is repolarizing the membrane. It becomes more polar again, and the interior of the axon becomes more negative again. Sodium gates become refractory, which means that the sodium ions can't enter the cell anymore. Potassium continues to leave, causing the membrane to return to a negative resting potential, and the sodium gates reset, and the potassium gates close. And by propagation, what I mean is the signal is transmitted down the length of the axon. So think of each one of these pluses and minuses as that little chunk of membrane that we were just looking at with the sodium and potassium ions opening and closing. That just happens bing, 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 all the way down the length of the axon. So the signal just travels. So all these gates are just opening and closing throughout, causing an electrical signal to travel all the way down. All right? So this is a busy diagram. Don't get bogged down with all the memorization of it. If you want to read it for your own understanding, that's great. Just know that the propagation of the signal occurs because of the ion channels opening and closing down the length of the axon. And the resting membrane potential is maintained by what we call the sodium-potassium pump. And so sodium ions within the cell fit precisely into the receptor sites on channel protein, and they basically are able to be expelled out of the cell and potassium ions into the cell. This is the opposite from what we were talking about when we were talking about the action potential. Potassium ions are normally going out of the cell and sodium ions into the cell. This one we're doing the opposite. This protein is maintaining the resting membrane potential so as to allow when a stimulus does elicit a massive change in electrical charge, an action potential can be carried down the length of the axon.